Hello viewers, welcome to this video lecture program. This is Ramakrishnan, Assistant Professor, Department of Biochemistry, St. Joseph's College of Arts and Science, Cuddalore. In today's program, I am going to discuss the topic DNA footprinting, a famous technique used to find out the DNA and protein interactions. Look at the pictures. In this picture, we will understand the concept of this heading clearly. First image represents the clean beach, that is, there is no footprints in the sand part. Then second, we will see some footprints. Then look at the third one, DNA molecule, pure one. Then the fourth one, DNA molecule with the protein, that the black spot. So symbolically represents that DNA is interacting with the protein, and the images like the footprints on the beach. So what are DNA and what are proteins, and why these two molecules has to interact? So in our system we have number of biomolecules. Among them, carbohydrates are the energy providers, nucleic acids are the information transport molecules, both instantly and generation to generation. Then proteins are the polymers of amino acids, which are the functional unit of all the characters. Now look at the DNA. DNA are the polymers of 4 nucleotides such as adenylic acid, guanylic acid, citric acid and thymidyllic acid, that is deoxyform. And these deoxynucleotides carries the information by two way, an instant one, that is expressing the gene. For example, synthesis of insulin from the DNA, that is by the way of transcription. First stage, DNA has to be converted to mRNA, then mRNA to protein. That protein is the functional form, that is for example insulin. Okay. Then second one, cell to cell information transfer, that is cell division. For example, in a birth, a child have the body weight of approximately 3 kilograms. After 10 years or 20 years, the body weight completely changing, that is increasing gradually and reaches the maximum. All these things are all because of cell division. And similarly, we will use some terms. A person is having baldy head or tummy. So by looking the person's father or grandfather, we will use to the conclusion, we will always use that yes, it is possible because his father is like that, his grandfather is like that. Okay, that is information from generation to generation. Okay, all these are all determined by gene. Genes are segment of DNA which code for the biologically active functional protein. But the gene never gets expressed without the contact of protein. That is our topic, that is DNA and protein interaction. In order to express the gene, DNA has to interact with the protein. Either it may be a replication or transcription, etc. All these things are all done by the combination. This will be determined by a technique, that is we are going to discuss today, called footprinting. Footprinting is the technique was developed by David Gallas and Albert Schmitz in 1978 and detected the DNA repressor gene in the leg operon. Actually, this technique is modified from the DNA sequencing technique invented by Maxam and Gilbert. In this technique, when we focus the principle, the gene of interest or the DNA will bind with the protein, and the protein will be protected for the enzymatic lysis, because DNAs are the enzymes that will cleave the DNA into fragments. It's called molecular scissors of DNA. So DNAs, that is deoxyribonuclease, is a endonuclease. This is a glycoprotein having the molecular weight of 39 kilodalton. It will specifically cleaves the DNA in the tetranucleotide sequence and generate 3' hydroxyl and 5' phosphate group segments. It will cut both the double helical DNA as well as single stranded DNA. Therefore, the DNA is converted into the fragments. That is first stage. That is the principle behind that one. And once the fragmented DNA is separated by using electrophoresis, polyacrylamide gel electrophoresis, and visualized. So we will get a ladder of DNA that will be sequenced and come to the conclusion where the protein is attached with the DNA. Okay. Next part, we are going to discuss the procedure. The major steps involved in this technique is, first stage is the collection of gene of interest, or the DNA, which has to be found whether the DNA is interacting with the protein or not, or in which part the DNA is interacting with the protein. Okay, so first stage is collection of DNA from the sample. Then second one, in order to find out the further studies, we have to amplify the gene of interest by using polymerase chain reaction. In this technique, that is in the footprinting techniques, we are using some other molecular tools. First one, this polymerase chain reaction. Second one, radioisotopic techniques. Third one, electrophoresis. We are using the three different fields in the one technique. Okay, so after the DNA isolation, the DNA is treated with a radio labeled compound, that is a radio labeling system, usually P32. Why we are using the radio labeling system? In order to find out where the DNA is present in the gel, that is detection part. One, we can use the radioisotope, or even we can follow some other techniques such as tagging with a fluorescent compound, or performing the third one, blotting techniques. All these techniques will help to find out where the DNA is located in the gel and the gel ladder appearance. The next part, this radio labeled DNA is treated with the lytic enzyme DNase 1 or deoxyribonuclease 1. This step will cut the DNA into fragments, and we will get the number of small fragments. So this digested DNA is used as a standard part for the activity of DNase 1, and another part from the PCR amplified DNA take and treat with the protein of interest, that is DNA protein interaction study material. For example, DNA polymerase, or DNA dependent RNA polymerase, or suppressor gene like repressor gene proteins. These proteins is treated with the DNA, and the next moment the DNA will bind with the protein and forms DNA protein complex. This is test. First we have taken and treated the DNA as such with enzyme, that is standard. This is test. Then after treatment with the protein and the DNA, or mixing the protein and DNA after a period of incubation, it is treated with the lytic enzyme deoxyribonuclease 1. Now the DNase will cut the DNA which is freely available, and the DNA which is bound with the polymerase or protein part will not be digested. Okay, it will be as such in the sandwich form. And next stage after the digestion, electrophoresis. The separated, that is digested samples are separated by using agarose gel electrophoresis. This electrophoretic separation gives the DNA in different ladder form. So that ladder will segment and gives a proper footprint region. So look at this image. First part is the pure DNA and enzyme. That is, we will see the clear ladder based on the molecular weight. In the second test, standard will give the clear DNA ladder. In the test sample, we will find the missing of some bands. That band part is called DNA footprint part. We will analyze this missing DNA footprint part by using DNA nucleotide sequencing systems. So after the sequencing, we will merge all the missing nucleotide and we will come to a clear conclusion where the protein and the DNA were interacted. Now come to the application part. DNA footprinting technique mainly used for finding the DNA protein interactions in order to find out the replication, initiation, elongation, termination, then transcription, initiation, elongation, termination, and the drug regulation part. So many synthetic drugs, the DNA interacting drugs that will be binding with the DNA and controls the regulation of gene expression. So that will be analyzed by this technique. And the next one, detection of hormone response elements and ligand binding. Hormone response element is the segment in the DNA that will be the region which controls the gene expression for hormonal action. Hormones are the chemical messengers which will coordinate the entire metabolic processes. For example, thyroid hormone. So are the steroid hormones. So these hormones, that is mainly steroid hormones, will bind with the DNA and forms a complex called a DNA hormone complex in the particular region called a hormone response element. That region will also be detected by using this technique. Thank you.